Hey guys, it's me. Hope you're having a great Monday. Uh, this is the beginning of your last week of new material. Um, we have 2 more days to finish up chapter 15, so that'll be today and tomorrow. Then on Wednesday um, and Thursday, you will be finishing up your last lab exercise 23, and then I think that's over the ear. And then on Friday, you'll have your last lab exam, which will be just over exercises 22 and 23, which will be the eyes and the ears and your senses of hearing and sight. Um, and then next week, I will be posting your last lecture exam at the beginning of the week, and then your final will be posted as well for you to have done by the end of the week. And then you will be finished with your dual credit anatomy class. Please let me know if you have any questions or need anything from me. Again, this week we are going to be finishing up chapter 15. You're going to be doing lab exercise 23, and you're going to have your last lab exam over lab exercises 22 and 23 to finish by Friday this week. So today we're going to be talking about the eyes and your sense of vision. And that is it's really pretty complicated, so we're going to have a lot of stuff to talk about today. Don't forget that you will have a quiz over chapter 15 as well, probably on Wednesday. And you'll also have a lab exercises 22 and 23 quiz probably on Thursday. So again, a couple more quizzes. We have to get those quizzes in there because if we're going to drop the quizzes, remember you can drop 1 lab quiz and 4 lecture quizzes. We have to get all of our quizzes in order for you to be able to drop those. And then next week I will ask for you to let me know which quizzes you want to drop. I will let you know which ones you have already, or how many you've already dropped, and then you can let me know which others that you want to drop if you have any left to drop. Okay, so as I was saying, today we're going to be talking about vision. Again, your visual system is what we're going to call this. Includes not only your eyes, but also accessory structures. We'll talk about some glands that are associated with your eyes, as well as your eyelids and things like that, your eyebrows, your eyelashes, things like that. Also includes your optic nerves, as well as your optic tract, and then sensory pathways that lead to the optic center of your occipital lobe of your cerebrum. One unique thing about our vision, our sense of vision, is that it can detect stimuli from a greater distance than any other sense that we have. Again, if you think about you're looking at the stars at night in space, you realize that light is coming from thousands of miles away from the Earth. So therefore, definitely our sense of vision can allow us to detect that light from a greater distance than any other sense that we have. We can't hear sounds from as far away, but we can see that light from that far away. Some of the accessory structures, as I mentioned earlier, would include your eyebrows. Again, our eyebrows not only provide shade for our eyes, but they also prevent sweat from getting in our eyes. Our eyelids, which are called our palpebra, with our conjunctiva, which are going to be layers that kind of go over the kind of thin layers that go over the eye. We'll talk about those in a minute. Or actually they're lined, the conjunctiva line the inner layer of the eye. You actually have a couple different layers. You have a layer lining the inner eyelid, and then you have a layer lining around the actual eyeball. So in our eyelids and our eyelashes, as far as function, help to protect our eyes from foreign objects getting in them. Obviously, they don't always work, but again, a lot of times when you have things flying in your face toward your eyes, you will blink, you will close your eyes, and those eyelids and eyelashes are helping to prevent things from getting in there, and the eyelashes actually, when you're blinking, are actually helping to knock things out of the way. You have the space between your upper and lower eyelid is called your palpebral fissure. The canthi are the actual corners of your eye. You have a lateral canthus, which is the lateral corner of your eye, and then you have a medial canthus, which is the medial or inner corner of your eye. And then at that medial canthus, you've also probably noticed a little mound, a little pink mound. It actually has a name as well. It's called the caruncle, and it contains modified sweat and sebaceous glands. So basically, you do have some oily secretions as well as just moisture. In addition to your lacrimal glands that are producing tears, you also have some oil being produced to moisturize the skin around your eyes, as well as modified sweat glands as well. You also have 5 layers of tissue that make up your eyelids. Of course, your skin is the most superficial layer. Under that, you have areolar tissue, which is that loose connective tissue. It's going to have some fat in it. You have muscle that also, obviously, you have that make up your eyelids, and that's what causes your eyelids to move when you blink. You have the tarsal plate, which I think is part of the. I'm not really sure what that is, but I was thinking. It's not really bone, but it's more of just like a stiff tissue that's helping to maintain the shape of your eyelid. And then you have your palpebral conjunctiva. As I told you just a few seconds ago, you have a couple layers of conjunctiva, and that palpebral conjunctiva actually lines the inner portion of your eyelid. So the palpebral conjunctiva would be the most deep layer of the 5 layers of tissue on your eyelids. So again, here we can see some of the major structures. Again, your eyebrows, your eyelashes. You can see your superior palpebral, and then your inferior palpebra. You can see the medial canthus with that small pink mound with the glands called the caruncle. You can see here's your lateral canthus, the lateral corner of the eye. The white part of your eye is called the sclera. We'll talk about that more in just a few minutes. The iris, again, the color part of the eye, and then the pupil is the black part of the eye, which is actually an opening into the inside of the eye that's covered by the cornea. Which is the clear outer coating of your eye. The clear coating of your eye that the light goes through is the cornea. The lens is a more solid structure that's further back behind that pupil inside your eye. Here we can see those 5 layers again. Here's your eyebrow, here's your eyelashes. You can see here we have the skin labeled. Under that we have that areolar tissue. Kind of looks like some fat in there. You have the muscle layer there. Then you have this tarsal plate here. This is the tarsal plate, again, maintaining the shape of the eye. And then underneath that you have that palpebral conjunctiva. I'm not sure if it's showing the other layer of conjunctiva here or not, but in any case, you do have the palpebral conjunctiva here. I guess we'll look at another picture here that will have the other layer of the conjunctiva. You can see the sclera here. You can see the lens here. You can see some suspensory ligaments. You can see the cornea here, the anterior chamber. This is the posterior chamber. The pupil here. So you can see lots of different things. And of course, here's your palpebral fissure. Again, the area between the upper and lower or superior and inferior palpebra, which are your eyelids. Okay, so moving on and looking at some structures, some more structures. The eyelashes, most people have a double or triple row of hairs in their eyelashes. Again, obviously, if you have a triple row, you're going to have those thicker eyelashes that ladies like to associate with more beauty, but some people only have a single or double layer. You have your ciliary glands, which are going to be the modified sweat glands in that caruncle, as well as at your eyelashes. Again, I guess kind of helping to secrete some fluid to kind of help keep you cool. And they do empty in the eyelash follicles. You also have some meibomian glands, not only in that caruncle, but also at the inner margins of your eyelashes that are producing sebum that are helping to keep your eyelashes moist, just like your sebaceous glands do for the hair on your body and your head and things like that. Again, the conjunctiva, we've already talked about the palpebral conjunctiva. Again, the conjunctiva in general are thin, transparent mucous membranes that help to lubricate the surface of the eye. You have a layer that is on the inner surface of the eyelid called the palpebral conjunctiva, and then the layer that's covering the anterior surface of your eye except over your pupil, so it doesn't go over the pupil, it doesn't cover that hole or that opening, that black portion of your eye, but it does cover the rest of the anterior portion of the eye. It's called the bulbar conjunctiva, and I'm not sure that it showed that in this picture. I was looking. Yeah, here it is. The bulbar conjunctiva again. So basically it looks like though that palpebral conjunctiva kind of curves around and becomes the bulbar conjunctiva on the anterior surface of the eye. And then all of this that is on the interior surface of the eyelid would be called the palpebral conjunctiva. Okay, moving on, the lacrimal apparatus. You actually have a lacrimal gland that produces your tears. We'll look at that in just a minute. It is innervated by parasympathetic nerve fibers from your facial nerve. So again, it is parasympathetic. and those tears are going to leave the gland through ducts called lacrimal ducts, and they're basically kind of more lateral. They're on the lateral portion, superior. They're superior lateral to your eye there. The superior and lateral portion of your eyelid is where they are, kind of underneath there. The tears leave the lacrimal ducts, wash kind of diagonally over the eye to lubricate it, and then they collect in the nasolacrimal duct, which basically connects your eyes to your nose. And so any excess tears will collect there and then which will then come out your nose. They'll empty into the nasal cavity and come out your nose. So this explains why when you cry, your nose runs, because those excess tears are going into that nasolacrimal duct and are coming out your nasal cavity. You produce about 1 milliliter of tears a day, again, barring that you are, crying a lot or something like that. So this is a graphic that really shows you how the tears are flowing. Here we have our lacrimal gland that is producing the tears. The tears leave through these little lacrimal ducts here, again, kind of the superior lateral portion of your eye. The tears diagonally kind of wash over the eyeball to keep it moisturized. And then they collect in these lacrimal canaliculi into the lacrimal sac, and then go down the nasolacrimal duct into your nasal cavity and come out your nose. So again, when you cry, you're producing excess tears, and so those are going to be coming out your nose, and that's why your nose runs when you cry. We also want to look, you guys, we have intrinsic and extrinsic eye muscles. The extrinsic eye muscles are skeletal muscle that actually moves your eyeball. The intrinsic eye muscles are going to be smooth muscle, and they are going to be constricting and dilating your pupil, which we'll talk about more in just a few minutes. So you have 6 extrinsic eye muscles that move your eyeballs themselves. 4 of them run straight from front to back of the eyeball. That is called anterio-posteriorly. So they run from the front to the back. So they run straight anterio-posteriorly from the anterior to the posterior of your eye. So, and there are 4 of those. You have the superior rectus, you have the you have the inferior rectus, which you can see here. So I think you can see. I don't like the anterior view as good as I do the superior view, or I'm sorry, the lateral view. The superior view allows you to see that superior oblique and superior rectus better, but the lateral view allows you to see all of them better, in my opinion. So anyway, we can see here the superior rectus. We can see the inferior rectus. We can see the lateral rectus, and then on the other side of the eyeball, we have the medial rectus. So here's our lateral rectus, here's our medial rectus, and then here's our superior rectus. And in this superior view, you can't see the inferior rectus, but in the lateral view, you can see the inferior rectus. So you can see 3 of the 4 of them really well. Here, you just can't see that medial rectus because it's on the other side back there behind that eyeball. But in any case, you can see how they run from the anterior to the posterior portion of the eyeball. So they are said to run anterio-posteriorly. And then again, the 2 that are positioned at an angle are called the superior and inferior oblique muscles. So here we can see the superior oblique muscle here. It's kind of medial to or deep to that lateral rectus. Or I'm sorry, it's deep to that medial rectus. This is going to be your medial rectus here. So the superior oblique actually looks like it's superficial to the medial rectus. So the medial rectus is more medial or underneath the superior oblique, and the superior oblique looks like it kind of runs through this trochlea and then up kind of maybe deep to that superior rectus. You can see again the superior oblique here. Again, it is running at an angle to the rectus muscles, and then you can also see the inferior oblique here. Of course, in the superior view, you can't see the inferior oblique, but here you can see that inferior oblique again on the bottom part of the eyeball with the superior and running, kind of diagonal to the eye and to the rectus muscles. The superior oblique running diagonal to the eye and the rectus muscles and on the superior portion of the eyeball. Remember that superior oblique is innervated by the trochlear nerve, and that's the nerve that allows you to look down. The lateral rectus, remember, is innervated by the abducens. Again, abd for abduct. It allows you to abduct your eyes or look from side to side. And then the other 4 nerves, the other 4 extrinsic eye muscles are innervated by your oculomotor nerve. Again, allowing you to move your eyes in all the other directions. Again, this is an actual picture from a cadaver. We see our eyeball here. We can see the superior rectus that is cut. We can see the optic nerve. We can see the medial rectus here, the lateral rectus here. Again, you can see the optic chiasm where the optic nerve from this eyeball and the other eyeball meet. Again, here you're not seeing that inferior oblique or that inferior rectus muscle. Okay, let's look at the eyeball itself. The eyeball is a sphere that is hollow and fluid filled, and it is composed of 3 layers or tunics. The fibrous tunic, which includes the sclera and the cornea, the vascular tunic, which includes the choroid, the ciliary body and the iris, and then the nervous tunic, which includes the retina. So again, fibrous is more about shape of the eye, vascular is more about blood flow and flow of nutrients, nervous is more about the actual sense of vision itself. So we will look at each tunic or layer of the eyeball. Again, we can see here the layers. Here, this is the liquid inside the eyeball itself called the vitreous humor in the vitreous chamber. Here we can see the retina or the nervous tunic is the innermost or deepest layer of the eyeball. The choroid or vascular tunic is the middle layer, and then the sclera or fibrous tunic, also including the cornea, is going to be the most superficial layer of the eyeball. And then we can see several of these other areas here too. Again, this is the vitreous chamber. We can see here the anterior chamber, which is between the cornea and the lens. Then, or I'm sorry, probably the cornea and the iris. And then the posterior chamber is going to be between the iris and the lens. So looking at the fibrous tunic, again, this is the most superficial layer or tunic of the eyeball. It includes 2 things, the sclera and the cornea. The sclera is the white of your eyes. It is the white layer, and it is pretty firm. Again, giving the eye shape, protecting internal structures, providing points of attachment for those rectus and oblique muscles. And it is continuous with the cornea. So basically, these are all part of the same layer. The cornea and the sclera are continuous with one another. The difference is the sclera is white, the cornea is transparent. What causes that is that the sclera is white due to large collagen fibers, whereas the cornea is more transparent because it contains smaller collagen fibers as well as proteoglycans and a lower amount of water. Again, the cornea, like the sclera, they are going to be more avascular. They're not part of that vascular tunic. They're part of that fibrous tunic. And again, we do have that transparent part of the fibrous tunic at the anterior portion of your eyeball called the cornea, because that is where the light enters your eyeball. Again, it is made of connective tissue matrix plus some stratified squamous epithelium. Again, it is transparent because it has a small number of it has smaller collagen fibers than the sclera. The sclera has larger collagen fibers. In addition, it also has proteoglycans and a lower water content. Again, the lower water content, in addition to the more proteoglycans, helps the light to enter the eye without being refracted. If there were water in if there was a lot of water in the cornea, then the light would be refracted or scattered or bent more, and so the light would not go through the cornea as well. It would be less transparent. As the light enters the cornea, it is bended or refracted to help focus the light on the retina. So we do want some refraction, but water would cause too much refraction and too much scattering of the light. And this is the only part of your eyeball that's pretty easily transplanted. Someone who has a damaged cornea, they can actually take a cornea from a cadaver and transplant it into that person's eye so that they can see. So it is the only part that's relatively easily transplanted. Okay, looking at the vascular tunic, again, this is the middle layer, and it contains the most of the blood vessels of the eye. It contains arteries that are basically branching off of the internal carotid arteries, and it does contain melanin, which makes it appear darker in color. There are 3 parts of the vascular tunic: the iris, the ciliary body, and the choroid. The iris is the color part of your eye. Again, the melanin in there causes it to have the color that it has, and again, the amount and kind of melanin that is produced is genetically determined, so that determines the color of our eyes. The iris is the structure of the eyeball that controls the amount of light entering the pupil, and it controls that by changing the size of the pupil. And again, this is where those intrinsic eye muscles come in. You have 2 groups of smooth muscle that are our intrinsic eye muscles that constrict and dilate the pupil. The sphincter pupilla are the muscles, the intrinsic eye muscles that constrict the pupil. They are innervated by parasympathetic fibers from the oculomotor nerve. So again, your pupils constrict when you're in that rest and digest mode because you don't need as much light entering your eye. The dilator pupilla are innervated by sympathetic nerve fibers from that oculomotor nerve, and they dilate the pupil. And this is going to be, again, when you're in that fight or flight mode, your pupils will dilate to allow more light to enter the eye so that you can see, let's say, to get away from danger. The ciliary body is another part of that vascular tunic. It is the anterior portion of the vascular tunic, and it produces the aqueous humor that fills that anterior chamber. Again, remember the anterior chamber is between the cornea and the iris. And the ciliary body is actually composed of a couple different things. Ciliary muscles that actually are attached to the lens and control the shape of the lens. Again, those ciliary muscles, like those sphincter pupilla and dilator pupilla muscles, are smooth muscles, so we do not have conscious control over changing the shape of our lens so that we can see. And then we have ciliary processes. The ciliary processes are connected to the lens by ligaments called suspensory ligaments, and these processes are the parts of the ciliary body that actually produce that aqueous humor. Again, that aqueous humor is just a fluid that is found in that anterior chamber between the cornea and the iris. And then the third part of the vascular tunic is the choroid. It is associated with the sclera, so it's going to be more attached to that superficial tunic, the sclera, or superficial part of the fibrous tunic called the sclera. And it is very thin and also pigmented because, again, remember, all the parts of this vascular tunic contain melanin, and so they are going to be darker in color. We can see a little bit better here. Here's our lens. Here are the suspensory ligaments connecting the ciliary processes and the ciliary body to the lens. Remember, it's the processes that are the actual part of the ciliary body that are producing the aqueous humor. And then we have the ciliary muscles that are contracting and relaxing to change the shape of the lens. So as these muscles contract and relax, then that is going to pull on these suspensory ligaments, which will change the shape of the lens. Here we can see the anterior chamber, not real well, but we can see it, again, between that cornea, and here is going to be our iris, which is the colored part of the vascular tunic. Here's the posterior chamber between that iris and the lens itself. Again, this is going to be filled with aqueous humor. Here we can see the lens itself. Again, you can see those suspensory ligaments connecting those ciliary muscles to that lens so that when they contract, it changes shape. We can also see the intrinsic muscles of the iris. When the sphincter pupilla contract, we get constriction. We get a smaller pupil, and then when the dilator pupilla contract, we get a bigger pupil, letting more light into the eye. So this is more sympathetic innervation of these muscles and more parasympathetic innervation of these muscles. Okay, the last part of the last tunic is going to be the deepest layer, which is the nervous tunic. It is the retina. It contains 2 layers. We'll talk more about these layers a little bit later. But the outer layer of the retina is pigmented, and it is composed of simple cuboidal epithelium. Pigments in this layer and the choroid actually help to separate sensory cells and reduce light scattering, and we'll talk more about that in a little bit. The sensory layer of the retina is called the neural layer of the retina, and it is going to be the innermost layer, and it contains the rods and cones, which are our photoreceptors. They are the sensory receptors in our eye that are sensitive to light, and actually in response to light, they are going to generate action potentials. When we look at the retina, there are some different things we want to talk about. This is a picture of the retina. The lens and the other parts of the focusing system. So parts of the focusing system include not only the lens, but also even the cornea and the aqueous humor in the anterior and posterior chambers, because all of those things refract light. They bend light as the light goes through them, helping to focus it on the retina. So when all of these parts focus light on the retina, they are focusing light on the macula and the fovea centralis. The macula is going to be right here. It doesn't really look yellow here, but it is a small yellow spot near the center of the retina. And then the fovea centralis is a small pit kind of adjacent to that macula, where most of the light is focused. So most of the light is going to be focused in that fovea centralis. It contains only cones, no rods, and these cones are very tightly packed, which gives it the ability to see very fine images, which means that it gives us the most visual acuity. When we talk about visual acuity, we're talking about the ability to see fine images. So because there are no rods here, only cones, and they are very tightly packed, the fovea centralis gives us the ability to see very fine images. We also have another part right over here of the retina called the optic disc, which is your blind spot. And I know it looks more yellow here, but it's actually a white spot. And this is going to be the area where the blood vessels enter and leave the eye, and also where the nerves there enter and leave the eye. And so there are no photoreceptors at all. There are no rods and no cones. And so any light that is focused on that part of the retina, you will not be able to see any images. So this is what gives you a blind spot, like when you're driving, because you have that area there, that optic disc, where there are no rods and cones, no sensory cells that are able to detect light. So any light that is focused on that area doesn't get turned into action potentials that your brain can interpret, so you can't see any images that are focused on that portion of your eye, and that's what gives you your blind spot when you're driving. Okay, looking at the chambers of the eye, we've already mentioned these, the anterior chamber, the posterior chamber, as well as the vitreous chamber. The anterior chamber and posterior chamber are both parts of what we call the anterior compartment. Again, these are anterior or in front of the lens, and they both are filled with aqueous humor. As I've already mentioned, the anterior chamber is between the cornea, which is, remember, the transparent anterior part of your eye, and the iris, which is the color part. And then the posterior chamber is between the iris, the color part, and the lens, which is inside your eye. Again, the fluid in these chambers, this aqueous humor, helps to maintain intraocular pressure, which kind of helps to also maintain the shape of your eye. It supplies nutrients to all the structures that are bathed by this aqueous humor, and it also helps to refract the light. So again, the aqueous humor in these chambers is part of the focusing system of the eye. And again, remember, it is produced by the ciliary processes, which are part of that ciliary body. Glaucoma is an abnormal increase in intraocular pressure due to the inhibition of circulation of this aqueous humor. So this aqueous humor is circulated, is returned from your eye to venous circulation through the scleral venous sinus. So if for some reason there's problems with this circulation, then it actually leads to glaucoma, which is just increased pressure inside of your eyeball because this aqueous humor is being produced but not circulating like it should. Again, then the vitreous chamber is going to be behind the lens. It's much larger than the anterior and posterior chamber, and it is filled with a different kind of humor. The aqueous humor is much more liquidy. The vitreous humor is more jelly-like, and it also, like the aqueous humor, helps to maintain intraocular pressure, help the eye hold its shape, and also holds the lens and retina in place, and is also part of the refracting or the focusing system of the eye. So again, the cornea, the aqueous humor, the vitreous humor, and the lens, all of these together help to focus the light on the retina. They help to refract the light so that it comes together at a point on the retina called the focal point. The lens itself is transparent. Now, if we were to look at it, at least in a dead animal, the lens looks more, not necessarily opaque, but probably more translucent. But in a living animal, it's going to be transparent, which basically means like see-through, like looking through a glass. And it is biconvex, meaning that it is convex on both sides. Generally, when something is convex, it's convex on one side, but on the other side, it's concave, like, let's say, a contact lens. But your lens itself is biconvex, meaning that it is kind of rounded on both sides. Its anterior surface is lined with simple cuboidal epithelial cells, and then its posterior surface is lined with long columnar epithelial cells called lens fibers. And these cells, all of these cells actually lose their nuclei and other organelles and accumulate proteins called crystallins that help to make this lens transparent. And it's also covered by an elastic transparent capsule, and it is suspended, remember again, between the posterior and vitreous chambers by those suspensory ligaments that connect it to the ciliary body and the ciliary muscles that allow it, and when those ciliary muscles contract, they pull on those suspensory ligaments and change the shape of the lens. Again, here we can see that vitreous chamber very large, again, posterior to the lens or behind the lens. Here we can see the anterior chamber between the iris here and the cornea, which is that transparent portion, anterior portion of the eyeball. And then here's the posterior chamber between the lens and the iris. Again, the iris is the color part of your eye. This is the pupil, the black part that you can see, but it's actually an opening where the light enters. When the ciliary muscles contract, it can either constrict this pupil, making it smaller, or it can dilate it, making it bigger to allow more light to go into the inner portion of your eyeball. Again, the functions of the eye, the main function of the eye is to focus light and then turn the sensory receptors in the retina, turn those light images into electrical signals that are then going to be carried to our brain by the optic nerves. When we talk about focusing, we're talking about converging the light. Again, refracting that light, bending it so that those light rays converge at a point that we call the focal point. Basically, as light passes through your pupil, it is focused on the retina by the cornea, the lens, and the humors. Again, all of those things refract the light, bend those light rays to focus them on the retina. Light striking the retina is then converted to action potentials, which are then going to be relayed to the brain. As the light rays pass through the cornea, the lens, and the humors, they are refracted and converge and cross at a point called the focal point. So the focal point is the point where light rays converge and cross. The lens can actually change shape to adjust the focal point on the retina. So you can actually have your lens, if the focal point is in front of the retina, the lens can change shape to focus it further back. If the lens is focusing behind the retina, then the lens can change shape to bring that focal point up on the retina. Sometimes, however, due to problems with those ciliary muscles, problems with our lens, problems with the shape of our eyeball, our lens, our focusing system is just focusing the light too far behind the retina or too far in front of the retina, and so we can't see well. And so that's why we wear eyeglasses or contact lenses, because those contact lenses can help to refract those rays of light or those images. Basically, what they do is they just move the focal point. They can move the focal point forward. They can move the focal point backward. Again, and convex versus concave lenses can move that focal point. One type moves it forward, one type moves it backward, and we'll talk more about that in a few minutes. So once the image has been formed on the retina, not in front of it or behind it, but on it, then it's going to be inverted. Then action potentials from this image are passed to the visual cortex of our cerebrum, which is in our occipital lobe, and there the brain will interpret those images, and it will turn the image right side up again. So the image is focused on your retina upside down, inverted, and then those signals, electrical signals from that image, are carried to your brain, and your brain is what flips the image right side up as it interprets those signals. So emmetropia is the normal resting condition of the lens. This is going to be when the ciliary muscles are relaxed and the lens is completely flat. When we talk about the far point of vision, we're talking about the point at which the lens does not have to thicken to focus. So basically, the far point of vision is how far away something has to be, and your lens can be in emmetropia. Your lens can be in its resting condition. The lens does not have to change shape. It does not have to focus. It does not have to thicken to focus that light. And so for normal individuals, that far point of vision is 20 feet away or further. So when things get closer to you, your lens, your suspensory ligaments or your ciliary muscles have to start contracting, and suspensory ligaments pull on that or push on that lens and change those and change the shape of that lens so that you can focus if it's closer than 20 feet to you. Further than 20 feet away, generally your lens can be in that normal condition. It can be in that resting condition, that emmetropia. The lens doesn't have to change shape or thicken to focus, so 20 feet away or more. Near point of vision is the point at which the object becomes blurry as it gets closer to you. So from 20 feet away, as things come closer to you, you can see your lens is going to have to be changing shape. Those ciliary muscles are going to be contracting, changing the shape of your lens so that you can focus, so that you can see those images. But, okay, so from your far point of vision, 20 feet away or further, your lens doesn't have to do anything. okay? But from 20 feet closer, your lens is going to have to be changing shape to allow you to see. But at some point, you're going to get to the point where it's going to be blurry. And wherever it becomes blurry, that is the near point of your vision. So the near point of vision is the point from 20 feet closer where things become blurry. It's the point between 20 feet to the point where your things become blurry. That's going to be at that distance, the lens is having to change shape. But at your near point of vision, the lens can't do any more. Okay, something comes close enough to your face that it is blurry and you cannot see it clearly anymore. Okay, that is called your near point of vision. So again, anytime an object is closer than 20 feet, the lens is going to have to change in shape. It's going to have to, the size of the pupil is going to have to change, and the distance between your pupils is going to have to change in order for you to be able to focus on that. And once you get to the point where it's right in front of your face and it's blurry, it's totally blurry and you can't see it clearly, then that is called the near point of vision. So the process of the lens changing in shape, so again, from 20 feet closer to you, the lens has to change shape so that you can see the object. The process of the lens changing the shape so that you can see the muscle, or so that you can see the object, is called accommodation. So your lens has to accommodate closer than 20 feet away from you. Any closer than that, the lens has to accommodate. It has to change shape so that you can focus on that object. And again, it is the ciliary muscles that are doing this. They are contracting, pulling on those suspensory ligaments, changing the shape of the lens so that you can focus on objects that are closer than 20 feet to you. That's called accommodation. And again, remember, those ciliary muscles contract due to parasympathetic nerve fibers from the oculomotor nerve, which is cranial nerve number 3. As accommodation occurs, the lens becomes more spherical, light is refracted more, and again, that is going to help you to focus those rays of light on the retina, even though this object is closer than 20 feet to you. Also, another thing that helps us to see is pupil constriction. As accommodation occurs, the pupils have to, the size of the pupils has to change. When our pupils constrict, it actually varies our depth of focus. Depth of focus is the greatest distance through which an object can be moved and still remain in focus. So again, as we're at 20 feet away from us, as we bring an object closer and closer to us, we are still able to focus on it because our pupil changes size, it constricts, and varies our depth of focus. As the smaller our pupil is, the greater the depth of focus that we can have. And it's also the reason why more light is required to focus, because again, if your pupil is smaller, you don't have as much light coming in. So you have to have more light in the environment around you as you bring things closer to you so that you can focus on that object that's closer to you, because the pupil gets smaller as things come closer to you to help you focus on that object. And so it requires more light for you to be able to focus on that object as it comes closer to you. The other thing besides accommodation and pupil constriction that's going to allow us to focus on things that are closer than 20 feet to us is called convergence. Again, our eyes have to move closer to each other, because remember, we said back here that when an object is closer than 20 feet, the lens has to change in shape. Okay, that's the accommodation. The size of the pupil has to change. That's the pupil constriction. The other thing that has to change is the distance between the pupils, and that is called convergence. As objects move closer to your eyes start to rotate medially. Again, we call it crossed eyes, but basically your eyes have to start rotating medially, bringing those pupils closer together so that the object is kept in focus as it is moved closer to you. And again, this is due to the to reflex contractions of the medial rectus muscle. So this is not something that you consciously think, oh, I've got to cross my eyes so that I can see this object that's right here in front of my face. That automatically happens. That is due to reflex contraction of that medial rectus muscle doing that. Again, so that medial rectus muscle is allowing you to cross your eyes, to bring those pupils closer as things come closer to your face so that you can see them. Okay, so looking at visual acuity, again, remember this is our ability to be able to see fine detail, fine images. Again, our ability to focus an image on the retina so that a clear image is perceived, ability to let us see clear images. Myopia is the technical name for nearsightedness. What this means is that you can see objects that are close to you clearly, but distant objects look blurry. Basically, the problem here is that your cornea or lens is too powerful, or else your eyeball is too long. And so what happens is the focal point is in front of the retina. And so how do we correct that? We use concave lenses, also called minus lenses. And those actually will push that focal point further back on that longer, that eyeball, or on that retina, because that eyeball is too long. If you have a very minor case of nearsightedness or myopia, this can actually be corrected by the LASIK surgery. LASIK surgery is, the actual name for that is radial carot. I don't know how to say that. Radial keratotomy, maybe. Okay. Hyperopia is farsightedness, and this is when we can see objects close to us clearly, objects further away from us clearly, but close objects are blurry to us. And this is because either our cornea or lens is too weak, or our eyeball is too short. So what happens is the image, that focal point is behind the retina. And so we can also correct that. We can correct that with convex lenses. We call these plus lenses. Okay? And we'll look at pictures of that. Right here we can see, in this case, either we have a problem with our cornea or lens, it's too powerful, or else our eyeball is too long, and so the focal point, the images are being focused in front of, here's our retina, the images are being focused in front of the retina. So how do we correct that? We use a concave lens. That concave lens pushes that focal point back on that retina so that we can see clearly. The hyperopia is just the opposite. This is farsightedness. So basically either our cornea or lens is too weak, or else our eyeball is too short, and so the focal point is moved back, and so the image is being focused behind, again, here's the retina, behind the retina. So we can also correct that with a convex lens. The convex lens will bend the light so that the focal point is brought forward, and the image is the light from the images is being focused on the retina. Okay, a couple of other problems with visual acuity, and this is what older people have, guys. It's called presbyopia. I have this. Basically, you have an increase in the near point of vision. So basically, as you get older, your near point of vision becomes further away. Okay? And so basically, this is due to the fact that your lens is less flexible, and so it doesn't do that accommodation as well. And so basically, if your near point of vision is further away from your face than 9 inches, you have presbyopia. And again, this is due to, the lens becoming less flexible, and so your accommodation power, the accommodation of that lens doesn't work as well. And so you're able to see things, like things that are closer than 9 inches are blurry to, than 9 inches are blurry to you. The average age of onset for this is the mid-40s, and basically you can correct this by wearing reading glasses or bifocals, also called progressive lenses. So like I have bifocals. So basically, guys, if you've ever seen someone who's older without their glasses on and they're holding things way out so that they can focus, it's because, again, remember, their near point of vision has increased. Their near point of vision, so the distance at which they can bring something closer to them and still be able to see it clearly has increased. So to be able to see something clearly, if they have it, 10 inches from their face, it's blurry to them. And so they have to put it further away from their face so they can see it clearly. And so those progressive lenses or those bifocals help to bring that near point of vision back closer to the eye so that they don't have to hold things very, very far out from them to be able to see it. And then the last problem I'm sure you guys have heard of is astigmatism. This is where the cornea and lens are not uniform, either the cornea and or lens are not uniformly curved, so light rays do not focus at a single point. Now, there's 2 types of astigmatism, regular astigmatism and irregular astigmatism. Regular astigmatism can be corrected with glasses with the opposite curvature, but irregular astigmatism is difficult to correct because it's difficult to correct with glasses. I don't know if they can do surgery or not, because there's abnormal curvatures of the cornea. There's no specific pattern of curvature. You can't really get a curvature of glasses to match that, so that you, or to go opposite of that, so that you can bring that focal point to where it needs to be on the retina. Okay, last thing we want to look at is the structure and function of the nervous tunic, which again is the retina. Remember, there are 2 layers of the retina. The neural layer, which contains 3 layers of neurons, You're going to have your photoreceptor cells, which are your rods and cones, your bipolar cells, and your ganglionic cells. And then within that neural layer, you have a couple of different plexiform layers, which are areas where these three layers of cells synapse with one another. So you're going to have an area where the photoreceptor cells are synapsing with the bipolar cells. That's going to be a plexiform layer. And then you're going to have a layer where the bipolar cells are synapsing with the ganglionic cells, and that's going to be another plexiform layer. And we'll look at that. We'll look at a diagram of that here in just a little bit. And then the other layer of the nervous tunic is the pigmented layer. And this is a single layer of cells. Again, it is pigmented because it has melanin. And this layer enhances your visual acuity by isolating the individual photoreceptors and reducing light scattering. So again, just allows you to be able to see images more clearly by isolating the photoreceptors from each other and reducing light scattering. So here's the diagram I was talking about. Here is our pigmented layer of our nervous tunic or our retina. And then all of this is our neural layer. Again, 3 layers of cells. The photoreceptor cells, these are our rods and cones. Again, the blue ones with the rod-like end are the rods. The pink ones with the cone-like end are the cones. Then we have the bipolar layer, and then we have the ganglionic cell layer. Again, the areas where these cells synapse with one another are called plexiform layers. So the areas where the photoreceptor cells synapse with the bipolar cells is called the outer plexiform layer, and then the layer where the bipolar cells synapse with the ganglionic cells is called the inner plexiform layer. Again, remember that our nose and our retina are the only 2 places in our body where we have these bipolar neurons. Remember, we said these are very rare, so this is the only other place besides our nose where we have these bipolar neurons. Again, the rods are, bipolar photoreceptor cells and they are responsible for our black and white vision as well as our ability to see in reduced light. They are found over most of the retina, but again, remember, they are not found in the fovea. You are going to have them in the macula, but they are not found at all in the fovea. And remember, they are more sensitive to light than cones are. So cones are not as sensitive to light. So therefore, we have to have more light to be able to use our cones. And we're going to see that cones are used for color. So this is the reason why if the light gets very dim, all you can see are shades of black and white. You cannot see color because the rods are very sensitive to light. The cones are not. So it requires lots of light in order to be able to use your cones. And since cones are responsible for color vision, this is the reason why it requires lots of light to be able to see color, and we can't see color if the light is very dim. The rods contain the protein rhodopsin, which is a purple pigment that changes shape when struck by light. When light strikes it, it separates into its 2 components, opsin and the photosensitive pigment retinal. And this photosensitive pigment retinal leads to hyperpolarization of the cell. Well, retinal, guys, comes from vitamin A. So this is the reason why, like, eating carrots is good for your eyes. Carrots have lots of vitamin A. Vitamin A can be converted to retinal. Retinal can be converted to vitamin A. In the absence of light, this opsin and retinal will be in the form rhodopsin. Light, when it strikes this rhodopsin, changes the rhodopsin into opsin and retinal, and it is the retinal that is going to lead to hyperpolarization of the cell. And you guys, these rod cells are very unusual because when they are not stimulated, they are depolarized. Remember, like with our muscle cells, when we stimulate, when the nerve stimulates the muscle, depolarization happens, and this causes contraction. Well, when these cells are not stimulated, they are depolarized. So in their resting state, they are depolarized. Okay, they're not polarized. They are depolarized in their resting state. And when light strikes them, hyperpolarization occurs. And this is what stimulates them. So basically, when the rods are exposed to light, remember, normally when a cell is stimulated, the sodium ion channels open and allow sodium to come in. Well, when these cells are exposed to light, sodium ion channels close. Okay, so again, it's opposite of what we have learned. With most cells, when stimulation happens, depolarization occurs. Sodium ion channels open and sodium comes into the cell. not with rods. With rods, when they are stimulated by light, the sodium ion channels close. So when they're not stimulated, the sodium ion channels are opening and sodium ions are coming into the cell. Okay, but with these, when they are stimulated, sodium ion channels close, and so less sodium comes into the cell. And what we're going to see is something called glutamate. The amount of glutamate decreases. Hyperpolarization decreases when light strikes the rods, and sufficient depolarization of the cells happens to release neurotransmitters that will then stimulate ganglionic cells. So we do end up getting depolarization of the ganglionic cells, but the rods themselves are hyperpolarized. And that is what causes them to release neurotransmitters to stimulate the ganglionic cells, which will then depolarize. So again, they're just a little bit unusual because depolarization doesn't lead to the release of the neurotransmitter. Hyperpolarization leads to the release of the neurotransmitter from these rod cells. Then that neurotransmitter will attach to the ganglionic cells, and the ganglionic cells will depolarize. Sodium ion channels will open, sodium will come in, and depolarization will happen. Also, these rods allow us to experience what we call light and dark adaptation. And basically, this is referring to the fact that the eyes adjust to changes in light. And this happens because when you have changes in the amount of light, you have changes in the amount of available rhodopsin. Again, bright light breaks rhodopsin down into opsin and what was the other one called? Opsin and retinal. Retinal, there you go. So when we have less light, we have less. When we have less light, we have more rhodopsin. Okay, when we have more light, we have less rhodopsin because light breaks the rhodopsin down. We also this light and dark adaptation also happens due to pupil reflexes. Again, bright light, pupils constrict to allow less light in. Okay, dim light, pupils dilate to allow more light in. Finally, light and dark adaptation happens because of changes in the level of photoreceptor function. Again, in bright light, rod function is going to decrease because remember our rods are more sensitive to light, and so they allow us to see in dim light conditions. In bright light, cone function increases. Remember I told you earlier that cones are less sensitive to light, and so they need more light to function. So brighter light, we're going to be using our cones more. We're going to have our pupil constricting to allow less light in, and we're going to have less rhodopsin, okay? And so that is what causes us to adapt to bright light. In dim light, we're going to have more rhodopsin. Our pupils are going to dilate to let more light in, and our rods are going to function more, and cones are going to function less. So those changes are what allow us to adapt to light versus dark conditions. Again, this is showing you the rhodopsin, and again, this is the retinal, and this is the opsin together. That's rhodopsin. Remember, in bright light, it breaks down, and it's going to be this retinal that is going to cause this G protein mechanism to function. This G protein is then going to produce CGMP, okay, CGMP, and then that CGMP is going to go over here to this open sodium ion channel, and it's going to close it. And that's going to cause hyperpolarization. And that is what's going to stimulate that rod cell to release neurotransmitters, which will then go across the synapse to the ganglionic cell. Those neurotransmitters will then depolarize the ganglionic cell. Sodium ions will come into the cell, and that will generate an action potential in the ganglionic cell. So the rod cell is not generating an action potential. The rod cell is releasing neurotransmitter that then binds to the ganglionic cell, and the ganglionic cell generates the action potential, which will then be carried to the brain. Again, this is just talking about what we just went through as far as the rod cell. In unstimulated conditions, you have the rhodopsin, the sodium ion channels are open, glutamate is constantly being released from the unstimulated cell, and that inhibits the bipolar cells from releasing neurotransmitters. So glutamate is keeping the bipolar cells from releasing neurotransmitters. When the rod cell is exposed to light, the rhodopsin will break down into opsin and retinal. This is then going to stimulate this G protein mechanism producing that CGMP. The CGMP will then go to the sodium gated ion channel, or to the gated sodium ion channel, I should say, and it will close that channel so that sodium can't come in, and so now the cell will be hyperpolarized when sodium stops entering the cell. Glutamate will be released from the rod cell. The glutamate will then allow the glutamate will then tell this bipolar cell to release neurotransmitters to the ganglionic cell. So again, basically in this, when the rod cell is exposed to light, hyperpolarization happens. The rod cell releases glutamate. Glutamate attaches to the bipolar cell, causes the bipolar cell to release neurotransmitters, and then those neurotransmitters will attach to the ganglionic cell that will be here across the synapse from the bipolar cell. And then that ganglionic cell will generate an action potential by depolarization. Okay, let's look at cones. Cones also are bipolar cells. They are more conical in shape. We saw those pink cells that look like a cone at the end. They are responsible for our color vision and visual acuity. So not only do they allow us to see color, but they also allow us to see clearer images. And again, they require relatively bright light to function. So again, in dim light, we don't have as much visual acuity because cone cells are not functioning as well. The rod cells are functioning more. So we can see in dimmer light conditions, but we can't see color, and we don't have as much visual acuity. Again, the cone cells are numerous in the fovea and the macula. Again, they're mostly there in those two areas. Again, remember that the fovea has no rods, only cones. As light intensity decreases, so does our ability to see color until all we can see is gray, because fewer cone cells are responding in dim light. Again, the cone cells are not as sensitive to light as the rod cells are. The pigment, remember the pigment in the rod cells is rhodopsin. The pigment in the cone cells is iodopsin. So I remember rhodopsin rod, RR. Rhodopsin is in the rod cells. The cones have the other pigment, which is iodopsin, which is retinal plus the photopigment opsin. There are 3 types of cone cells, blue, red, and green. Each iodopsin, so there's different forms of iodopsin. One that allows us to respond to blue light, one that allows us to respond to red light, and one that allows us to respond to green light. So there's different forms of iodopsin, and each one of these forms responds to a very narrow spectrum of wavelengths of light. So again, that's the reason why we have one that responds to these blue wavelengths, one that responds to red light wavelengths, and one that responds to green wavelengths. But you can see there is some overlap, especially with the red and green, but there's a little bit of overlap with the blue and the red and the green as well. And so since they overlap, this allows us to interpret a gradation of colors. And there are several million gradations of colors that we can interpret because of the overlap of the sensitivity of these different forms of the iodopsin. Not only do we have 3 different forms of iodopsin that are sensitive to wavelengths of blue, red, and green light, but we also have different forms of each one of these. We have different forms of blue iodopsin, different forms of red, different forms of green. So, for example, there are 2 forms of the red iodopsin pigment that allow us to see, that allow different people to see different reds. So you can imagine, it's kind of neat to think about how this color of, let's say, candy apple red that I see, it may not be the exact same color of candy apple red that you see, because I could have a different form of that red iodopsin than you do. So there's different forms of those. Not only are there 3 different forms as far as responding to different wavelengths of light, but there's also different forms in different people of those 3. So it's really neat to think about how we all don't see things the same way. We all don't see the same colors. Of course, you already knew that because you know people that are different forms of colorblind. But again, this is the reason why we have different forms of colorblindness. Different genetic mutations can cause, differences in these different forms of these iodopsins, which cause different forms of colorblindness. It's kind of neat to think about. Again, rods and cones aren't evenly distributed across the retina. Cones are more numerous in the fovea. Again, there's no rods at all in the fovea, only cones, and even in the macula. And again, remember, both of these areas of our retina are associated with visual acuity, associated with us being able to see clear images. And rods are more sensitive over most of the rest of the retina. So you have more rods over most of the retina except for the fovea and the macula, and you have more cones in those. And in fact, the fovea has no rods at all. Okay, so looking at the inner layers of the retina, the rods and cones synapse with bipolar cells that then synapse with ganglionic cells in all areas except the fovea. Except in the fovea, the ganglion cell axons converge at the optic disc and then exit the optic nerve, and then those impulses travel from the optic nerve to the visual cortex and the occipital lobe. But in the fovea, we have the highest visual acuity because there is an absence of ganglionic cells and there is a greater concentration of cones. So again, that fovea gives us the highest visual acuity because not only do we have no rods and the greatest concentration of cones, but also we have an absence of ganglionic cell processes. So again, we have very little convergence with the cones because basically you have one cone cell synapsing with one bipolar cell, whereas with the rods, we have higher levels of convergence or spatial summation. We have one bipolar cell receiving input from numerous rods, one ganglionic cell receiving input from numerous bipolar cells. So we have numerous rods sending a signal to one bipolar cell. We have one bipolar cell sending a signal to we have several bipolar cells sending a signal to one ganglionic cell. So we have a lot more signal coming in at the same time, is what I think of it as with our rods. But with our cones, we call that convergence, when you have lots of signal coming in at the same time. With our cones, we have just one signal coming in at a time, and so we have little convergence. So we have much more visual acuity. We're able to see much more detail. Not a lot of light sensitivity, but lots of detail that we can see with those cones because we have fewer signals, less information coming in at the time, so we can get a much clearer image. With the rods, to me, it seems like we have a lot more information coming in, and so we don't get as clear of an image. We're a lot more sensitive to the light, but we don't get the clear image. With the cones, we're less sensitive to the light because we only get just a little bit of information coming in at a time, but we're getting a very clear image because we're getting a small amount of information at a time. Okay, looking at receptive fields. Receptive fields, again, are the area from which the ganglionic cell receives input. So again, a smaller receptive field, we're getting less input in. Again, more visual acuity. With a larger receptive field, it's a larger area, which means we're getting more input in, which means, a larger receptive field, we're getting more visual acuity. A smaller receptive field, we're getting less visual acuity. Those in the fovea centralis are going to be smaller than other parts of the retina. And again, that really doesn't make sense because the fovea centralis is giving us a lot of visual acuity due to the absence of ganglionic cell processes. So to me, I would think a smaller receptive field will give you more visual acuity. So I think I may have just typed that in there wrong. The fovea centralis has the smaller receptive fields, and so therefore we're getting more visual acuity. Two types of receptive fields. The on-center ganglion cells generate more action potentials when light is directed onto the receptive field, and so we get more intense. We respond more to the intensity of the light. So with the on-center ganglionic fields, we're responding more to how bright the light is. With the off-center ganglion fields, we get more action potentials when light is off or when the light does not hit the center of the field, and so it's going to allow us to respond more to contrasts in light. So with the on-center ganglion fields, we are responding more to how bright the light is. With the off-center, responding more to light versus dark. Also, interneurons in the inner layers modify the signal before the signal leaves the retina. Again, we have horizontal cells in the outer plexiform layer that synapse with photoreceptors and bipolar cells, and then we have amacrine cells in the inner plexiform layer that synapse with bipolar and ganglionic cells. So remember that outer plexiform layer is where the rods and cones synapse with bipolar cells, and we're going to have horizontal cells in that layer. The inner plexiform layer is where the bipolar cells synapse with ganglionic cells, and we're going to have amacrine cells in that layer. And then we also have interplexiform cells. These are cells that form that are in the bipolar layer, and they synapse with amacrine, bipolar, and horizontal cells to form a feedback loop. And these different interneurons, again, these are horizontal cells, amacrine cells, and interplexiform cells. These are all interneurons. They enhance borders and contours. They increase the intensity at boundaries, such as the edge of a dark object against a light background. So they allow us to be able, they form this feedback loop and allow us to be able to see borders and contours and the difference between light and dark better, to allow us to see edges of things better. So if we go back and look, I think this diagram that we looked at earlier had, it has here's an interplexiform cell that is synapsing with both ganglionic cells and with the horizontal cells. Here's an amacrine cell that is synapsing with in this layer, in the interplexiform layer. Here we have that horizontal cell that is synapsing in that outer plexiform layer, and then we have this again, these are interneurons. Here's one. Here's the type, the horizontal cell. Here's the amacrine cell, and here is the interplexiform cell that is basically synapsing. It's kind of creating this feedback loop between the all three layers, as well as the horizontal cells and the amacrine cells. It's creating a feedback loop that is allowing us to see the edges and the contours and the borders of objects. Again, just giving us, to me, more visual acuity there. So what is the pathway that light takes from our eye to our brain? Well, first of all, obviously, those sensory receptor cells, the rods and cones, are going to be receiving the light itself. And then once they release those, once they stimulate those bipolar cells to release neurotransmitters, those ganglionic cells, so the rods and cones will stimulate the bipolar cells to release neurotransmitter. The bipolar cells will release neurotransmitter that will then bind to the ganglionic cells and cause depolarization and generate an action potential. This action potential will then travel along the optic nerve to the optic chiasm. In the optic chiasm, this is some interesting information as well. In the optic chiasm, some axons cross to the opposite side of the brain. Some do, not all, but some, and that results in both hemispheres receiving visual input from both eyes. So again, that's responsible for our binocular vision, which we'll talk about in a minute. But the action potentials go from the optic chiasm to the optic tract, then to the thalamus. Remember, all senses except the sense of smell go through the thalamus. Also, not only do they go through the thalamus, but they also go through the superior colliculi of the midbrain. If you remember, the midbrain, that superior colliculus, receives the auditory and the visual inputs that cause certain reflexes. Remember, when we see something, it causes us to turn our head toward it. Okay, so that reflex, the reason why we are able to, we turn our head toward that, bright light or something that we see, is because these action potentials are not only going through the thalamus to the visual cortex and the occipital lobe, but also going through the superior colliculi of the midbrain to cause that reflex to happen. And then this is all just talking about how those inputs come in, and they come in from our visual field, and they cross over to both sides of the brain in this optic chiasm, so that we have visual input from the visual fields of both eyes crossing over in that optic chiasm. You know, some of it stays on that side, but some of it crosses over to the other side. And this gives us our binocular vision. Areas seen with both eyes at the same time due to an overlap of those visual fields and that crossing over of the information from the left to the right side. The visual fields partially overlap, yielding depth perception. So again, this binocular vision is what gives us our depth perception, which is our ability to distinguish between near and far objects and to judge their distance. So again, if you're blind in one eye, you don't have binocular vision, you do not have depth perception. You can't tell how close or far away something is. Again, these are just some of the eye disorders. We've talked about the myopia, the hyperopia, the presbyopia, and the astigmatism. Some other things that can happen, retinal detachment. If the retina was to detach from the optic nerve, then you can go totally blind. We talked about glaucoma. Cataracts, basically the lens just begins to cloud. Macular degeneration, this is common in older people. My father-in-law actually had this. It can result in the loss of acute vision. So again, you may not may what you're seeing. You may have a black spot in the middle of what you're seeing. You can only see the peripheral. I think is what that is. And then of course, diabetes causes decreased and problems with circulation to the eyes, which can result in loss of vision as well because it damages. Because that damages to lack of blood flow to the eye can damage those rods and cones. It can cause death of the cells due to lack of oxygen and nutrients to those cells and to those parts of the eye. Okay, please let me know if you have any questions. I know this was kind of long. Some of it can be a little bit confusing. On Tuesday, we will be looking at the ear, and then we will be finished with all of our new material in this class. Well, not all of it, because you're still going to have to do lab exercise 23 on Wednesday and Thursday, but you're going to be finished with all the lecture after tomorrow. So again, please let me know if you have any questions, and we'll talk to you later. Bye-bye.